Okay, so how do we go about recording or measuring the dependent variable? Well, I've made a little smart art thing here that is not completely complete, and I'd like to point out is these little categories are not independent of each other. So let me talk about that. So correctness. We can measure the dependent variable as how accurate you are in your response. Maybe it's a test score on some kind of, vocabulary test or something like that. Rate is another measure. How many times you performed the behavior during some specific time frame. Frequency, how quickly did you do it? Now you see, like I said, these are not necessarily independent of each other because, of course, you're going to do it faster. Usually, if you do it faster, it's probably going to produce more behaviors within a time frame, right? So you can see how rate and frequency are tied together. I would never ask you a test question that asks you to distinguish among these finely grained measures. I'm just trying to familiarize you with some terminology. Degree, the intensity of responses. So there are different ways that we could assess that. In the field of embodied cognition, there's this new push to assess anger using some kind of degree of response kind of measure. And so, for example, if I do something to make you angry and they give you an opportunity to play a challenging game with this phantom opponent who's supposedly in the other room, you get the opportunity to try and blast your opponent with white noise in the speakers that they're wearing on their heads. What I would be measuring as the dependent variable is how loud you turn that white noise up to blast him with. That would be a degree measurement. The amount would be the total number of responses, and again, you can see that the amount might be tied to frequency and rate, right? It also, might be tied to correctness because maybe I'm only going to count up the correct responses. Latency, how long does it take you to start responding? And then duration, once you start responding, how long do you spend doing it? These are, like I said, not even a comprehensive list. I could come up with more. They're tied together. The idea is there are a lot of different ways to measure the effect of the independent variable on some dependent variable. Now here's an interesting question. Should you include more than one dependent variable in your research? Absolutely you can. You can collect as many dependent variables as you want. But again, I'm talking about experiments here. I'm not talking about other kinds of research. In experiments, you really only want to collect the dependent variables that you plan to use and that will aid in your understanding of the effectiveness of the independent variable. So only do it if it's going to add to your understanding of the independent variable, and only do it if you have a plan for how you're going to analyze that dependent variable. A lot of the research that you're reading in the literature may have multiple dependent variables. I was just reading a study the other day that had 8 or 10 dependent variables. I don't know how they got these participants to participate in this study. They studied them over a 3-month period and gave them all these different tests. And frankly, I have to tell you, for me as the reader, they had me at one test. They really didn't need to do all those dependent variables. But I think they felt like their case would be stronger about the effect of the independent variable if they included more dependent variables. And they knew how to analyze it, and they had an intention for why they included all those DVs. So it made sense in that study. Like I said, you may notice it in the studies that you're reading also, that a lot of times they have more than one dependent variable. You don't want to be like this guy though. Get all the information you can. We'll think of a use for it later. Your dependent variables should always be tied to some kind of hypothesis that you've already asserted. And so the opposite of testing a hypothesis would be to collect a bunch of dependent variables and figure out what to do with it later. That's not really how you're supposed to collect data, especially not in psychology. I was just talking to somebody who's a biologist, and they were talking about, well, a lot of times I do, DNA research, and it doesn't become clear why I'm doing it until, I've already collected the data. Well, that's fine and stuff, but you got to remember with psychology, we're asking participants to do stuff for us. And, like our field loses credibility if we're asking people to do things and even we don't know why we're asking them to do it. You want to have a strong intention, a really clear experimental design before you start collecting data from human subjects. Okay, so how do you decide on a dependent variable? Well, you want to make sure that you're using good measures. So what does that mean? It should be a valid measure of whatever it is that you're assessing. Remember, we talked about what validity is before. It should be directly related to the independent variable so that changes in the independent variable have a chance of affecting the dependent variable, right? So you want something that's sensitive to the independent variable. You want something that clearly will measure the effects of the independent variable. So for example, if your independent variable is instructions to learn, then a good dependent variable would be a memory test that would reveal whether you learned or not, right? If your independent variable is exposure to songs or no songs, a test of the content that you supposedly studied while exposed to songs or not would be a good example of a dependent variable. If the independent variable is whether the material was presented in handwriting or it was presented in typewritten form, then a good dependent variable might be the accuracy of recognition of the words that are being conveyed. It needs to be reliable as much as it can be. Of course, this is farther down on my list because, people vary from one day to the next as far as their attention span, their motivation levels, and so independent variables might have a certain amount of power, but so do extraneous things that you have no control over in your participant. So the dependent variable might produce different scores on different days for the same individual because of things that the individual brought with them to the testing situation. So as reliable as can be expected is probably what that should say. All right, now nuisance variables. Nuisance variables are any unwanted variable that is popping up in our experiment and interfering with our method, our design. Nuisance variables tend to cause the variability of scores within groups to increase. So what that means is, for example, within the 60 frame rate condition, a nuisance variable would cause a wider range of responses than you would normally expect. It tends to increase the spread of the scores within a distribution. It doesn't usually cause the distribution to change its location on the chart. So let me give you an example of what I'm talking about here. When I was a grad student in Albuquerque, I tried to use a demonstration of the serial position effect that works great anywhere else I've ever taught. When I taught in Sacramento, it worked great. It works great up here because for most participants, most students in an intro to psych class, they don't really have the names of presidents memorized. When I was teaching in Albuquerque, though, I got a really weird set of memories of presidents' names, and it took me a while to realize that it's because in Albuquerque there's a quadrant of the city where all the streets that are going north-south for several blocks in a row, for about 25 blocks in a row, they're named sequentially after presidents. So it starts with Washington, then it goes to Adams and Jefferson and blah blah up to, I think it ends with Hoover, I think, might be Eisenhower. I think it's Eisenhower it ends with. So I get this list of names that most people can't remember on a list of presidents' names, but these students in Albuquerque were able to produce a really odd rate. And that made me realize that, of course, there's a nuisance variable present. There's a quadrant of the city with the names of presidents written on it. So here's an example of what effect that might have. So you've got the range of the red columns is showing us people who maybe memorized the list. And you'll see that there are more people who are remembering more presidents in the memorized condition. fewer people who are remembering so many presidents in the minimum American history condition remembering any presidents, but the distributions are still in the same basic area. The most commonly recalled number of presidents is around 11, no matter what your experience is. It's just that more people are remembering around 11 when they had memorized the list than are remembering around 11 when they had minimal American history. You get more variability between people when there are nuisance variables present. And that can cause you to draw incorrect interpretations of the effectiveness of the independent variable. You might think that whatever you've done didn't matter, when in fact it might have mattered, except for that you've got this weird variability among people because of a nuisance variable that you didn't know about. Sometimes people call these nuisance variables extraneous variables, and I've already talked to you about extraneous variables. They cause confounded experiments, so they're called confounders. So let's talk about these nuisance variables that we call extraneous variables. Any uncontrolled variable that can cause unintended differences between groups would be an extraneous variable. Any uncontrolled variable that can cause unintended differences between groups. So it could be things like a power outage that occurs during the testing of the control condition, but not during the testing of the experimental condition. I actually had this happen in my research back in the olden days. We used slide projectors instead of having a computer to present lists of items one by one. And my slide projector, periodically, the light bulb would blow out, and I'd have to dismiss my participant who was sitting there doing the experiment until the light went out. And luckily for me, at that time, I was doing my participants one person at a time. And so if the light bulb blew out, I would just dismiss them. But imagine if I had decided I was going to do the entire experimental condition in one sitting and the entire control condition in another sitting, and the power had gone out for the control condition and not for the experimental condition. Now you've got a different experience. Something different has happened for one group that didn't happen for the other group, and it could serve as a nuisance variable or an extraneous variable. It could be a confounder. I'm purposely using all these words interchangeably because your stats professors will totally do it. Maybe the researcher is friendlier to the control participants than to the experimental condition, and so you end up with the two groups scoring differently, not because of the independent variable, but because of the way that they were treated. All right, let's go ahead and take a little break and we'll talk about confounding some more.